already. So in this second session, we first have Anat Ben-Zvi, Anat Ben-Zvi. Oh,
Yeah.
Ben-Zvi, oh, never mind. She's a professor in life sciences, working, wait, I have to, this is a word I learned yesterday, proteostasis. Yeah, it's probably a very interesting topic that we would love to learn at some point. But she works a lot with worms. So she's going to tell us about the rare diseases and worms.
Okay. So thank you for sticking with us for the second part. I am actually Uta's neighbor, so she comes by my office a lot. And she was really insisting that I should participate, and I kept saying that I don't know what I have to contribute because I don't do applied science. It's not that I have anything against applied science, but it seems like all my questions seems to be basic, and even when I want to think about it, somehow I don't get there. But Uta is very convincing, and so I have agreed, came up with a title, and then came up with this talk, which I hope you will enjoy, which talks about my perspective on my journey in doing basic research and how it speaks to applied science, or how is it shaped by also application and thinking about application, even though on an everyday basis I don't think it does, but then sort of thinking about it, realizing that it does. So I kind of went back to my history, and I am in my training a biochemist, and I used to use a very basic system to ask my questions.
You need to talk to the microphone.
Okay, so I used to use very basic system to ask my questions, and the reason for that is because, as Sebastian said, I am very much interested in protein folding, and I was completely intrigued by a family of proteins called chaperones. That what they do is help other protein fold or reach their three-dimensional structure. So basically, if you think about the protein problem, we have a protein that is being produced as a polymer. Okay, we already heard about polymer, and now it's a line polymer that needs to get to a three-dimensional structure, and that process needs to be assisted by many other factors. One of the main factors that helps in the cell is called chaperones. And so I was really intrigued by those machines. I really wanted to understand how they are working, and that's what I spent most of my PhD doing. And I like that system very much because it's a very reductive system. I would have a test tube, I would know exactly what I put there. That will be like this is this amount of salt, this is this amount of ATP, this is the amount of the protein, and then I could devise or understand and study those machines. And so most of my PhDs, I spent time doing this type of curves, which will be kinetics or will be concentration-dependent behaviors of how chaperones interact with their substrates. And every time we will come up with like interesting insights on the system and write this paper. We'll be really enthusiastic, and we want to send it in one of those meaningless journal like Science and Nature. And when we do that, we will always get the question is, what is the physiological relevance? I mean, very nice work, beautiful work that you have done, but what is the physiological relevance of what you have done? Okay, so towards at the end of my PhD, thinking about this system and really coming with this idea of a new way of thinking about how chaperone can take this protein aggregates, which is when protein go bad and they create these structures that are not specific but very strong, and you want to take them out, and we explain how the chaperone are using anthropic forces to actually untangle this protein. Again, what's the physiological relevance? And then we said, okay, maybe this is relevant to other systems, and we got it into, okay, you can talk about this chaperone doing those so many different functions in the cell, and not just working with aggregate, but it also help transfer protein from one area of the cell to another. And we suggested and showed very nicely, both computationally and experimentally, that this system is associated again with this anthropic pooling, and got really, really excited, send it to those really nice journal, and again would ask what is the physiological relevance. I would say that we managed to climb all the way from Journal of Biological physiological chemistry to a proceeding of the National Academy of Science, but still not science paper. So at this point, I got the message, okay? And when I was trying to look for a postdoc, I was trying to look for a more physiological relevance. And I will also say, I did not put her in this presentation, but she has a very a strong impact on me. So my grandmother kept saying, this is very nice what you're telling me, I don't understand anything, but how are you going to make me young? Because that is what I care about, and if you're doing science, you have to think about me and my health. And so, with coming to think about what am I going to do in my postdoc, I realized that I have to look at the physiological relevance, and I realized physiological relevance have two very important axes. One is, the more you go towards human, the more physiological relevant you are, okay? So, obviously, if you're in a test tube, that is not very relevant, and if you're using yeast or shrimps or other things, nobody's going to spend a lot of money to get your genome, but if you are human, then there's going to be physiological relevance, and then immediately you're going to get recognition, and that will impact the way where you can actually publish your science. I have to say, I had to decide where I put my boundaries. So worms is where I got. I didn't go much far in this axis. But then the other part of the physiological relevance, the physiological relevance axis is the difference between physiological and disease. In fact, the further you go from physiological and more into the realm of the disease, the more physiological relevance you have, okay? So if you're actually doing basic research that is linked to disease, then you can explain your research, you can sort of think about it, and so I realized that I have to have physiological disease, and I just talked about protein aggregates, and to me protein aggregates were this huge mass of proteins that will clump in a test tube, or for you, is if you take an egg and you boil it, that's aggregate, okay? But in disease, that's diseases like Alzheimer's, Parkinson, or Huntington, where you can find these plaques that are linked, or mainly one protein, that are associated with the disease. And so now I have physiological relevance, and I can do my research, and this is why I wanted my own computer, and hopefully this will work. So this is physiological relevance. This is a worm, okay, that is expressing human disease, and now it's barely moving, and you can actually track in real time the protein aggregates, those little dots. Those are in real life, looking at a microscope, looking at protein aggregates. And when I went to this lab, I was like, ooh, moving test tube. Okay, the biochemist in me was very, very enthusiastic about this system. And so now, finally, we managed to get to the science. We could convince people that what we're doing is really relevant and physiologically relevant to actually publish. And we can show that when you have one protein in the cell that is aggregating, it's actually impacting the quality control, those chaperones that I studied many years, and those chaperones are now a resource that proteins compete for. And then if one protein is aggregating, other proteins that need those chaperones will not have enough, and so they will crash out of solution, and we basically get a snowball of aggregation. Okay, so we can show this interaction, and that was really, really nice, and even got perspective of how it impacts and the way we're thinking. I have to say, sometimes I'm much more proud of my JBC paper than this paper, but yeah, again. So here I am, starting my own lab, and what would I do? Basic science, because this is with the physiological relevance, because I already understood it. If I want to publish well, I have to do it. And so what I basically do is I study protein folding in a multicellular organism, or proteostasis, which is protein homeostasis, and we put those questions into an organism. So the same question I asked in test tube, how those chaperones assist other proteins to fold, I now ask in a multicellular organism. So I ask if I need to make a muscle, and muscle is built out of this huge protein complexes called sarcomere, which are more than 200 proteins building something extremely accurate. And then this thing is also doing work. Do you need the same quality control for this system where you need for, let's say, neuronal sending signals where it's receptors and neurotransmitters and so on and so forth, which is a very different type of proteins. Are protein the same because proteins are always built from the same structure, or because they're doing different things, they require different chaperone system? If you want to take the analogy, can I take a Kia Soul, my Kia Soul, to the Mercedes-Benz garage, will it get the service it needs or not? I mean, those are two cars. What is the difference between them? And so we did that. We did some link to aging, because aging is very interesting and brings you a lot of attention. So again, and we looked at signals, looked at switches, and looked at protein development and differentiation. And basically, mainly in the realm of basic science, if you think. But then, as I said, Ute made me think, so I started to ask myself, do I really do only basic science? And here I need to switch a little bit my hats from my research lab to things I do in my spare time, which I don't have, but I still do. And one of the things I do in my spare time is help other people write grants. I usually do it with young researchers. I also do it with the Research and Development Center in the Alavai's area. And what I do is I explain to new researchers, how do you need to write grants? And for example, in the research and development, I work mostly with young researchers that are doing mostly applied sciences, and we're trying to think, how do you write an ISF grant? Okay, so the first thing we discuss is basically, what is the difference between fundamental, basic, and applied research in writing grant? And when you think about the basic research, the idea is that basic research is finding something new. Okay, you're looking for something that is unknown, you're building new knowledge, you're extending the knowledge or the ground we're standing on by extending knowledge. So it doesn't have to have immediate practical application, it focuses on the mechanism. Usually when I have an hypothesis, and it's like, it might have something applicable in the future, but right now we're asking a question and we're interested in that question. Whereas applied research, in many ways when you're talking about it, you're talking about, okay, I have the knowledge, using the knowledge that I have, how am I going to use that knowledge towards an application? Now we already saw in the first two talks that it's true, but it's also not true, because application drives basic science and basic science right application. Sometimes you are standing on known knowledge, but you need to develop new knowledge in order to actually answer some of the application. But one of the interesting aspects of this is then, if you look, for example, at the most basic research, and we were told here in the first session that we are so lucky here in Israel because we have funding that is completely basic science. The Israeli Science Foundation, they make you write a statement that explains how your research is basic. So you have to convince them, really truly, that it's basic. And one of the researchers that I worked with on writing an ISF grant just got her grant rejected because it was too applied. Okay, so you have to be very basic. And yet, when you look at what they ask reviewers, there are 4 questions that you have to answer, 25% each one of them. 25% is who is the researcher, basically what have I done in the past to afford me this, now giving me more funding. 25% is what I actually suggested, so only 25% is the project I'm actually suggesting, okay? 25 is originality and innovation, which is what we call, okay, the groundbreaking aspect of this proposal. And then 25% is importance and implications, okay? So if you cannot convince that your really, really basic science is important and applicable, then you have a problem. And that's 25%. 25% can grant you, instead of an O or an A, and a B or a C, because, you know, whatever you're doing is not necessarily important in the eyes of whoever is reviewing this grant. So you have to convince that it's important. And how do you convince that something is important? You go to the physiological relevance, okay? So you go to the human and you go to the disease. You go back to basically implying that although you're doing basic research, it has an application, and no worry, maybe not now, but give me 10, 20 years, I will actually produce an application to whatever I'm doing. This is the way we are getting funded, and as I had a discussion in the break, funding is mostly what we're doing. We're busy and trying to get those grants. And you can see that already in those applications, if you go with projects that are low risk, okay, or have low gain, meaning you don't convince that they actually have an implication, those are batch projects, those are incremental. This is how it's defined. So you have to work here, you have to go to the groundbreaking, yes, but you also have to sort of produce something that is affecting other people. So I sort of, thinking about it, realized that, yeah, I do basic research, but I also do basic research apologizing. And I always have to somehow convince people that it really, really is good to pay for my research because maybe down the road there will be applications. So that is one way I wanted to sort of think about this problem, but then I wanted to approach it really and look back at what I'm doing and ask myself whether or not I really do basic research. And as I showed you right now, one of the things that is sort of shaping the way I do my research is funding. Okay, so if I look at significance and importance and implication, then it shapes the way I do basic research, even though it's sort of basic. If I keep looking for availability of funding, and in funding research is changing, and I'll give you the best example. So Ministry of Science used to have grants where you will be scored, and 20% of the score will be the potential of implication. And that potential of implication could not necessarily be immediate, okay? In the last round of application for the Ministry of Science, you had a 3-year grant, 2 years worth for doing basic research, but the 3rd year will only if you have an application and you had to show how you're going to either commercialize it or make it benefits to other people. So in that very basic grant that you're getting, you're already required within the 3 years of the grant period to bring the product or something that has impact on the general product. So, for example, I have a grant with Loeb that we used to build an AI machine, okay, and validate that machine in order to study aging. And our product is a web server where you can look your favorite genes and ask whether or not it's relevant to aging. Okay, so we have to give something sort of back, and again, it's not necessarily immediately. The second thing that shapes the way I do science is collaboration. I love to do collaboration. I simply enjoy it. I keep joking about the fact that I'm the only worm lab south of Jerusalem. So if you want to do worms in this area, usually you come to me, and so that makes me do all kinds of really weird stuff with other people. So, for example, my worms tend to be an object of, let's say, interest. So one of the things that we have been using is using them for all kinds of application of microscopy. So this is working with someone in engineering that builds his own microscope, and we wanted to use this microscope in order to actually follow up cell division in an embryo in C. elegans. So I took the worm, I cut them up, I got the eggs out, we did all kinds of other stuff. We got this working. This is very nice study. People really like to kill my worms. So people want to test all kinds of substances and see whether or not they're toxic or whether or not they reduce toxicity. And so we have a collaboration with, again, engineering or sometimes with the chemistry department, throwing all kinds of stuff at the worms and asking whether or not worm survival is now affected. This could be done directly if you want to test a compound, or let's say you would have a compound, both of those examples are compounds that are supposed to affect bacteria, and worms eat bacteria, so you try to see whether or not the bacteria are now more toxic, and so you can use worm in order to do that, very effective. And then, let's say more fruitful and fun collaboration for me is working with Esti, which is a system biologist, but also very much data-driven and AI-driven, and really using her knowledge to ask questions in the system. And what we do here a lot with Synergy is that I use my system to test predictions she gets in her system, and she uses her tool set to help me ask the question I need to ask when it comes to big data or things that I don't have availability to do. And so this is, for example, one Synergy like that, where we did an analysis on huge data sets in human, looking at the expression of chaperones. I told you they're coming back across tissue, and we could show back to the question, the different tissue has different expression patterns of chaperone, same chaperones, but expressed at different levels and different patterns, and we can show that that pattern is relevant and conserved all the way to worms. So same chaperones that are upregulated in muscle in human are also upregulated in muscle in worms, and not only that, those chaperones are actually linked to disease of muscle. So using this type of tools to do this sort of analysis. The last sort of area that I want to go to, and how basic science is sort of sometimes meeting more applied science, is kind of a weird area, and that is the realm of rare disease in general. So rare disease are this really interesting type of disease where you have really few individuals that are actually sick, okay? And the problem when we look at those systems is that there is no incentive for the industry to study or develop drugs or treatment for those diseases, because the disease that I'm going to talk about has 15 patients in Israel. Okay? You are not going to spend millions of dollars to get a drug for 15 people. Okay? Yet, those are devastating diseases, and you want to study them, you want to understand them. And so, often, I find myself sort of being pulled into those things. And what's interesting is when you go to those consortium, many times what you have those clinicians, so the doctors that actually treat the patient, basic scientists. Usually the basic scientists know a patient. So either know a patient or know someone that knows a patient, not further than that. Otherwise, what are you doing there? And then you have parents and advocacy groups that try to get funding in order to actually to do this research. And we had this is a really interesting disease called a protein called the Ikashi. And we had a meeting, and that meeting were all these various people, and really any basic scientists that started talking said, well, I had a student in my lab and her son was sick, or I had a student in my lab and she has a friend that has this. So you really get to those diseases like this. And this is one example why I got sort of dragged into this. There were 15 patients in Israel, 9 died, all of them are dying because of heat shock. Okay, I study chaperone and heat shock. Okay, so, and what's really interesting is that this Ikashi protein that was associated with the disease, what it's actually doing is taking one of the chaperone I talked before, and take it from the cytoplasm, the main area of the cell, and put it in the nucleus, another area in the cell. So that's all it does. It's like a carrier. It takes the protein from one place to another, and then we have this really devastating disease. We don't understand why. I should say, we're not going to say how many years I'm doing chaperones. I didn't know that HSP17 has this carrier. So this, it has the mutation is conserved, and this is what you can do with glowing worms. So we can actually have a mutation in the gene in C. elegans, and now you can see that the circle, that's the nucleus, and in the wild type you can see there's GFP inside the nucleus. Basically the protein goes in, and in the mutation the GFP is remaining outside. So we develop a model, C. elegans. Now we can ask questions where we can go back to this. So if I sort of take a look at this, so we have the funding that can shape science, we get collaboration that shapes science, and we have sometimes simply urgent need that can shape science. So, sort of like meeting a problem where you really want to help, and there is no financial gain there, but you just want to lend a hand and use your science for good. And I will conclude again with what Gabi said before, the most important thing is people that do the work. And so those are my lab members and my collaborators and the funding. And thank you for listening.
Okay, we have a couple of minutes for questions. Must I ask a question?
The clear paradox between signing. Okay, the question is like this: how would you define? Is there a non-applied implication? Okay, something which has a very broad influence on only pure science, right? Maybe that's what ISF is kind of looking for, because there's this tension.
Yeah.
I'm looking for advice.
Well, I don't know. I have to say the only time I did something that actually had no implication was when I was a PhD student and really stupid, and there was this essay that wasn't working for me, and I spent like 3 weeks trying to figure out why. I did. It was absolutely useless because it was basically some contamination that was working inside. So yeah, so sometimes you ask those type of questions. I don't know. I mean, I agree with you in many ways. And this sort of presentation forced me to look at this way, that I consider myself very basic science, and my question starts with how protein do their exercise, okay? But then as I was going through graduate studies, those diseases of protein misfolding started coming up, and then all of a sudden chaperones, that was a field dying. Basically, if you look at publication, you would see like there was a boost. It was really, really hard because heat shock response was the first transcriptional response discovered, so it was the first real system where you can understand how genes are regulated. So there was like this boost of studying, and then it started going down, and then they found protein misfolding disease, and like, ooh, we're going up again, okay? And so, and then you go down, and then there's this boom of aging, and then again we're going up. So I agree with you that it also drives this. But then again, I mean, CRISPR is one of those examples where you went there because you want to understand how bacteria fight viruses, and then you end up with gene editing that will impacts everybody's work here. I mean, I use gene editing to that mutant that you saw in C. elegans was a gene editing. Okay, so it's like immediately transferred. So sometimes it does this, and sometimes it's like more. It takes 20 years to figure out why. Yeah.
Short question thank you for this great talk, Anat. I even understood.
Trying.
No, the question, is there really no major journal where you can publish research without physiological impact, without being asked for that?
There was this boom of PLOS ONE, I don't remember how many years ago, where the idea was if you did the science and you did it rigorously, it should be published, okay? And for a while, this journal even was Q1, and people will publish there, and then it's disappeared. Now it's like Twilight's Q1, okay? There are some other journals that have this type of ideas, but I don't think I've ever seen a journal that was really, really clear from this. Normally, what you would say, they will tell you, they wouldn't tell you now it's not no physiological relevance. They will tell you it fits more a specified journal. So basically, go to the journal of worms eating this, and this, and what happens to it, kind of. Okay, so they basically, if you want to talk to the broad audience, and you want to have visibility for your research, you have to have those physiological application. Otherwise,
I mean, when I published my master's thesis, that's ages ago, in biology, we wrote at the end, or I wrote at the end, this may be relevant for this, but there was only one sentence. The whole research was completely basic, But now you are asked to really do research that is.
Yeah, and my PhD was like that. But you are going for a postdoc. You need a position. You want a position. You cannot publish in JBC and get a PI position. You have to strive for the Science, Nature, Cell, whatever journals that are the key that unlock the PI position. And so you confirm to the fact that you have to ask questions that will allow you to get those journals and publish in more probability. Let's say it's not impossible to get there, but it's very hard. It's not very likely.
Okay, thank you. Now we have